everyone. Thank you for joining session 3 of the workshop today. So just real quick, we wanted to remind everyone that this will be recorded, and once the annual meeting has completed its sessions, we will be throwing up all the recordings onto the CDRC website, so you can go back and listen to them again. Our agenda and speaker bios will also be located on the website. You can actually access those now. Just a helpful tip that they are located under the resources tab if you're looking for those items. We also wanted to remind everybody that if you have any questions, please feel free to submit them into the Q&A box, which you can find to the left of your screen, or I'm sorry, right of your screen. And then also, if you're interested in joining the Collaboratory, you can email us at cdrc@cpath.org if you're already not involved. And if anything pops up or you need any assistance with anything, then you can contact Kitty Boji, and her email is up here on the screen as well. So I will go ahead and pass it off to Marco.
Thanks, Pam. And I'd like to welcome everyone to this session 3, and we got through the first day of our annual meeting. It was the rare disease day, so we had a lot of discussions on rare diseases. But we're transitioning over now to special populations. My name is Marco Skeeto. I'm the executive director of the Cure Drug Repurposing Collaboratory. We're a public-private partnership along with the FDA. Who have been funding all of our initiatives to date so far to move this effort forward on repurposing. We also work very closely with NIH, primarily NCATS, through the CDRC and the Cure ID platform. I have a brief introduction on our effort. If you're not familiar with it, that's on our website as a presentation, and I'll point you into that direction if you want some more information. So CDRC is part of the Critical Path Institute. We're a nonprofit organization out of Tucson, Arizona, and we primarily work in this collaborative effort with a number of different stakeholders, but the primary ones that we really tend to gravitate towards is industry and the regulatory bodies. But we obviously have a huge interest in including patients and researchers and clinicians and all the other groups that make up the difference in moving things forward from a regulatory perspective as well. So, obviously it's in our name, real world data and drug repurposing. And we're right now really interested in learning how to be able to use real world data in order to generate hypotheses on efficacy of drugs primarily, and using those existing drugs for new indications, because we really think that we need to maximize the utility of those drugs that we already have in our armamentarium. And we do this by a number of different ways. Cure ID is one of those ways in which we bring drugs in that have been used in different ways, basically clinician reports. But we're now exploring other ways in which we can do this: electronic health records, partnering with registries. Those are other sort of data sets that can help inform and provide additional data for us to evaluate. And that's primarily going to be a discussion for tomorrow. But it actually obviously impacts a lot of the work that's ongoing, both now within what CDRC is doing and with what other groups are doing here as well. But it's really then the potential to transform that real world data into real world evidence, to be able to utilize that to potentially supplement the sort of the next steps that would go. So potential of increasing the likelihood to inform clinical trials and to increase the likelihood of clinical trials actually being performed using repurposed drugs. So I'll stop there. I'm really interested in looking forward to this discussion. We've got a large, one of the largest panel discussions that we've had for this annual meeting. We've got 12 groups here, so I'm expecting a lively conversation. We've got a good group of individuals from a variety of different backgrounds. talking about a number of different things, and special populations is a key area that we want to pursue further. But I'll stop here and let Matt and Kate introduce themselves, and then the panelists.
Yeah, thanks, Mark. I really appreciate that. My name is Matt Lawn. It's a funny last name. It's pronounced like on your front lawn, but spelled like laugh on, is what I say, and have said it for my entire life, as most of my family members do. And I'm a neonatologist at the University of North Carolina in Chapel Hill, and really have had the pleasure of working with Marcos Quito and others at CDRC to help work on this really important panel today to talk about special populations, of which neonates is one of them, certainly not the only one. And we have a really great presentations this morning. I'm really looking forward to them. And with that, I do clinical trials mostly for my research and my academic pursuits. And I will turn it over to Kate.
Hi, everyone. My name is Kate Burkowski. I'm an ORISE Fellow with the Office of Medical Policy at the FDA. I also work part-time as a oncology clinical research nurse at the NIH Clinical Center. And my focus at the FDA is on pregnancy and neonatal sub-projects. So I'm really excited for the discussion today. We'll have each of the panelists briefly do an introduction. So Anoop, do you want to begin?
Sure. Good morning, everyone. I'm Anoop Chawla from Vanderbilt University and Medical Center. I serve as director for the Modeling Adverse Drug Reactions in Embryos Research Network. Modeling Adverse Drug Reactions in Embryos is a mouthful, so we go by MADRE. And we're a public-private partnership, NIH funded as well as foundation funded, focused on trying to improve the pace and throughput of drug development for diseases particular to women's health, to pregnancy, and to trying to stave off adverse outcomes upon drug exposures during pregnancy to neonates. And so we do this leveraging pharmaceutical engineering approaches, human-inspired engineering frameworks, and other types of applied science techniques that integrate real-world evidence and really try to integrate phenotypic reproducibility based on clinical phenotypes at early stages of drug discovery, and look at how we can trace best practices that work to augment drug discovery all the way from the bench to clinic and then into post-market surveillance. So kind of a broad catchment area for our research network, but certainly drug repurposing falls squarely within those interests. And we're very keen to try and identify best practices for leveraging big data to systematically identify ways that we can repurpose drugs to then integrate into clinical practice to address many orphaned diseases of women's health and pregnancy. In terms of CDRC, I'm very happy to serve as co-chair of the Special Populations Coordinating Committee, which is the host for this morning's meeting. So very glad to have everyone on the call today from that angle. And I also work closely on several of the Collaboratory's COVID-19 projects. So I'll stop there, but very glad to be participating this morning. And thank you to all the fellow panelists for joining. Looking forward to a really great discussion.
Thanks, Anoop. Rachel?
Good morning. My name is Rachel Greenberg. I'm a neonatologist at Duke University, and I spend much of my time as a clinical researcher with the Duke Clinical Research Institute. I do a mixture of clinical trials and epidemiology type studies, and one of my roles at the Duke Clinical Research Institute is oversight of the day-to-day activities of the Pediatric Trials Network, which is an NICHD-sponsored trials network under the Best Pharmaceutical for Children Act. And our focus is to study drug product formulation, drug dosing, efficacy, and safety, as well as device validation, with the ultimate goal of improving child health. And one of the main ways that we are one of the main ways that we're mandated to do that is by updating drug labels to reflect appropriate dosing and indications for children, including infants, which is my clinical specialty as well. So thank you for the opportunity to be on the panel, and I'm looking forward to the discussion today.
Thanks, Rachel. Next is Millie.
Good morning, everyone. My name is Millie Dugal. I'm a staff fellow in the Office of Medical Policy, CDR at FDA, and I've been a Cure ID team member since 2016. My background and training is in maternal and child health, and hence my interest in drug repurposing for pregnant women and children. My role within Cure ID is to help expand our work into special populations and, with the team's help, identify sub-projects that would benefit pregnant and neonatal populations. I had recorded a talk for today's panel, which is, as Marco and others pointed out, it's on the website. If you've not had a chance to, look at it, my talk was focused on the need to develop survey tools to address some of the challenges that are posed by drug repurposing in special populations. I discussed how when Cure ID started to expand into special populations, we wanted to focus specifically on infectious diseases in pregnant women, especially those diseases that have the potential of transmission of infection to the fetus. And that's when we began to have conversations with experts and stakeholders about congenital CMV. It impacts both mother and the neonate, and there's nothing approved for use in the perinatal context. I also discussed the need to develop survey tools to gather information from clinicians, in this case using Cure ID, that can help us better understand the landscape and the variability of the management of congenital CMV. Thank you so much for having me on the panel, and I'm really looking forward to discussing how we can all work together to help these populations.
Thanks, Millie. Next, we have Matt Robinson.
Hi, yes, thanks for inviting me today. I'm an assistant professor in the Division of Infectious Diseases at Hopkins. My research interests prior to COVID were primarily in diagnostics for acute febrile illness and evaluation and management of drug-resistant gram-negative infections in India, particularly in the neonatal population, where we had the challenge of evaluating the comparative effectiveness of different antibiotic treatment strategies using real-world data for neonates with gram-negative bloodstream infections. With COVID, I have turned some focus to using the electronic medical records in the US to create machine learning models for clinical prediction to be used at the bedside for illness severity, and also using large real-world data sets to evaluate treatments that are used for COVID for their effectiveness. Thank you.
Great, thank you. Next, we have Kaiser.
Hello, everyone. My name is Kaiser Aziz. I, too, am an assistant professor here at Johns Hopkins. I'm a neonatologist, so very excited to see all the other neonatologists on this call, this truly is a special population. And then my primary focus is on neonatal severity of illness, where I'm using real-world data, real-world experiences to bring clinical data together, because one of the primary barriers and factors, problems that we all face is that we just don't have data harmonized at this point. And I'm doing this data harmonization through the Odyssey OMOP model, where as the director of our Neonatal Precision Medicine Center of Excellence, we're using some of the lessons learned through COVID to build some of these national registries. Again, just thank you for giving me the opportunity to be here today.
Great, thank you. And next we have Perdita.
Good morning. I am Perdita Tila Zapata. I am a program official at the National Institute of Child Health and Human Development, specifically in the obstetric and pediatric pharmacology and therapeutics branch, where you can tell from our name, our focus is broad from maternal, fetal, pediatric pharmacology. And our research is in those areas broadly. My particular portfolio includes clinical trials and pharmacoepi research. And my primary day job is to oversee the Best Pharmaceuticals for Children Act clinical program at the Institute, where we prioritize drugs that need to be studied in the off-patent space. We sponsor clinical trials through the Pediatric Trials Network that Dr. Greenberg mentioned earlier. and we disseminate that data to the public via our coordinating center. So it's my pleasure to be here, and thank you.
Thanks so much. Next, we have Kanwaljit.
Thank you. Hi, everybody. It's good to meet you. My name is Kanwaljit Singh. I'm the Executive Director of International Neonatal Consortium here within our INC, within CPATH. It's just like CDRC, INC is another public partnership, or public-private partnership within CPATH, and we, as by the name, we focus exclusively on neonatal conditions. It's a public-private partnership of diverse stakeholders consisting of industry members, academic researchers, nurses, families, regulators, with our ultimate mission of accelerating unmet drug development needs in neonates. We operate in a pre-competitive space as a pre-competitive collaboration to address the measurement and assessment of clinical outcomes in neonates, and also basically what we do is we focus all of our efforts on some of the most common conditions that you will find in a neonatal intensive care unit. The reason I'm actually here on this panel is just like everybody else on this panel who has an interest in real world data. INC is also big time into real world data, and we're actually already started working on an FDA approved funded project that involves collection and aggregation of real world data along with clinical trial data to answer some of the most unmet, questions in the neonatal space. First one we're working on is bronchopulmonary dysplasia. We're trying to collect data from electronic health records, registries, BPD clinical trials, and try to construct a disease progression model for BPD. Then we're also focusing on collection of laboratory routine lab values from NICUs, and basically trying to use aggregated NICU data to create generalized database reference values for all commonly used lab values in the neonatal population. Obviously, there are more projects that we're working on, but these two are the ones that we have most common direct, connectivity with CDRC, and the hope is that we will basically in the near future we'll be able to collaborate with each other and rather than basically competing with each other. So it's nice to be here today, and I look forward to the conversation. Thank you.
Thanks so much. And next we have Prabha.
Hi, good morning, everyone. My name is Prabhavaswanathan. I'm a pediatric infectious disease specialist, and I'm a medical officer at FDA, CDER, in the Division of Antiviral Products. Our group is, our primary function is to regulate the development and optimization of antiviral drugs for all antiviral diseases. So my portfolio is quite broad, but my personal interests lie in maternal fetal medicine, the care of neonates and pregnant individuals. And my research initiatives really are focused on addressing areas of unmet medical need for children, pregnant individuals, and their babies. Part of that work is Congenital Cme, which I had the good fortune of being plugged into this group to see if we can gain some traction and move the ball forward in the space. So thank you so much to the meeting organizers for allowing me to participate today, and I'm looking forward to the discussion. Thanks.
Thanks, Prabha. And next we have Ginny.
Hi there. This is wonderful to hear about all of the other panelists. I'll tell you a little bit about myself. My name is Jenny Taylor. I'm a neonatologist assistant professor at the University of North Carolina at Chapel Hill. I did record a presentation, so encourage everyone, if you haven't had a chance to access that, to review it. It focuses on one of my research interests, which is the development of a neonatal global rank score. And that is, instead of most clinical trials that use composite endpoints in research, such as death or BPD or death and another outcome, to use a tool called the Global Rank Score that's been used quite a lot in the world of adult cardiovascular clinical research, and take that into the neonatal world, where different endpoints can be incorporated and ranked by severity. So improving clinical relevance of the endpoint, as well as improving efficiency and power of the endpoint. And so in that talk, I discussed those things more in depth, and also the development thus far, which has included key stakeholder engagement through a modified Delphi consensus process, and we'll use real world data in the development of this global rank score. So thank you so much for including me today, and I look forward to hearing from everyone.
Thanks so much. Next we have David.
Hi, everybody. David Kimberlin. I'm a professor of pediatrics at the University of Alabama at Birmingham, where I'm co-director of the Division of Pediatric Infectious Diseases. Since the mid-90s, I have led the first collaborative antiviral study group, now the Congenital and Perinatal Infections Consortium, research efforts in congenital CMV and neonatal herpes and infantile influenza, neonatal enteroviral sepsis. The congenital CMV studies are, I think, what are relevant to our conversations here this morning. Beginning in the 1980s, the CASG, the Collaborative Antiviral Study Group, evaluated intravenous ganciclovir in babies with symptomatic congenital CMV disease, and they looked at two different doses. The higher of those doses was taken into a randomized control trial conducted in the 1990s. That's when I joined the group and began leading those efforts. It was a phase 3 randomized controlled trial. Half of the babies got IV ganciclovir for 6 weeks, half got placebo. In the 2000s, we did a PK/PD study of oral Valganciclovir in the same population, symptomatic congenital CMV infected babies, and established the dose of Valganciclovir that gave the same systemic exposures to ganciclovir as did intravenous ganciclovir. And then most recently, in terms of completed randomized controlled trials, we compared 6 weeks of antiviral therapy with 6 months of antiviral therapy, again with symptomatic congenital CMV disease. All of the studies I just described started therapy within the first month of life. We also have very recently completed a study called Valgan Toddler, where we started treatment beyond the first month of life. Unlike the first 4 trials where there was either virologic or clinical evidence of benefit, for the Valgan Toddler study starting beyond the first month of life, we saw virologic benefit, but we did not see clinical benefit, suggesting that treatment needs to be started in the first month of life. And now we have a large biomarker study that is going to launch soon, and other natural history studies that have been conducted over the years as well. I very much appreciate the opportunity to be part of this group and the panel today, and look very much forward to the conversation that we will be having over the next hour and a half or so.
Thank you so much. Next we have Jason.
Good morning, everyone. Thanks so much to the group and to Marco for the invitation. I'm Jason Lang. I'm a pediatric pulmonologist by clinical training. I'm at Duke University. I'm particularly interested in clinical trials and special populations and epidemiology. My specific areas are pediatric asthma and bronchopulmonary dysplasia. I wear a couple of hats here at Duke. I'm the pediatric PI for the American Lung Association's asthma clinical research centers. Actually, it's airways now, in addition to asthma. So focused on finding new and better ways to treat pediatric asthma and early childhood wheezing illnesses. And then my other area of interest is working within DCRI, the Duke Clinical Research Institute, with Rachel Greenberg and others, in ways to treat and prevent bronchopulmonary dysplasia. I've been the PI for a couple of the PI of the Clinical Coordinating Center at Duke for a couple of the Pediatric Trials Network trials in that area. I did record a talk for this that sort of merges kind of two of my areas of interest, repurposing a currently approved drug for children, potentially for the use in neonates at risk for BPD. So just one potential example of repurposing drugs for a really high risk and at need population. So looking forward to participating.
Great, thank you. And last we have Barbara.
Hi, everyone, and thanks so much for including me. I lead Cures Within Reach, which is a different from most of the other speakers. We focus solely as a nonprofit on finding and funding clinical trials, testing already approved therapies and new indications. We're disease agnostic, institution agnostic, and geography agnostic. Most of the funding we provide is what we call come back when funding. We're funding the pivotal first in human study that, when successful, provides the data needed for follow-on funding and follow-on trials, whether it's from the NIH, industry, private donors, and private investors to get involved. We currently have 33 ongoing clinical and preclinical trials around the United States in 21 different diseases at 28 different institutions, not just the US, but 7 other countries. And 24% of those ongoing projects are in the pediatric space. When we think about special populations, it's not just pediatrics, as most of the people here have experience in. We look at special populations slightly more broadly, including not just pediatrics, but we have a 2021 initiative in veterans and military, and also a very large effort this year we launched in the diversity, equity, and inclusion space, including health inequities, including underrepresented researchers based in the US, racial and minority researchers that are not getting the funding at the same levels as other populations. And then finally, underserved researchers in low and lower middle income countries. So happy to be here and look forward to the conversation. And I will add, in closing the panelists' intros, that it's quite appropriate that this topic is today, since today is World Prematurity Day. So thanks.
Oh, that's great to know. I didn't know that. Thank you so much. And thank you for all the panelists for being here. Like, we have such an amazing group. Matt, do you want to start off with one of our discussion questions?
Sure. So the first one would be, how can real-world data, real-world evidence be used to inform clinical repurposing trials and help a repurposed therapy become standard of care? What can the CDRC and the repurposing community do to bring the vast number of AI results for repurposing opportunities into human clinical trials, not just earlier risk mitigation? And Kate, are we going to have presentations as well?
We have the recorded presentations. I'm not aware of anyone doing any specific presentations, unless Marco, do you know?
No, I think that this is really going to be a panel discussion about certain topics, and I think that this was one topic that we were thinking about discussing. And we've heard, you know, a really great overview of all the panelists. And, you know, actually, I'd like to just, you know, I think that this also can be a little bit of a free-flowing discussion about things that do come up. And Barbara, I think, you know, although you're a little bit different on the panelists, but, you know, this is the nice thing about having these types of panel discussions, is bringing in other viewpoints and other groups that are doing things. And although we're having an interest in neonates and pregnant women, I've always been thinking of, you know, what other opportunities are out there as well to look at special populations. And you mentioned a few of them. You know, veterans come to mind. There are, you know, a number of others that you mentioned as well. But I'm wondering with the veterans one, are you working closely with the Veterans Administration to do work like that?
Our effort launched actually on Veterans Day 1 year ago. So we just had our first anniversary, I guess, and announced plans to continue this in 2022 for our Veterans Initiative. Looking at impacting veterans issues, we are actually selected this year projects in PTSD as well as traumatic brain injury. We are engaged with a number of different stakeholders around veterans to help spread the word about the funding opportunity. We worked with a number of organizations, all the Veterans Association, veterans research teams across the United States. There's a initiative called NAVREF, which is the National Association of Veterans Research Foundation, I think is what it is. There's 80 some odd research institutions connected with veterans hospitals and veterans administration facilities across the United States. There's an organization called Friends of the Veterans Administration, or FOVA. We partnered with them to, again, help spread the word. The other thing we're doing is using not just the Veterans Administration and those connected with it, but also veterans and active military to help review and prioritize and help us select the projects, because it's not just projects to impact veterans, but we also want to get veterans and active military engaged and involved in which projects we should select. And that's a really important part of inclusion, is having people at the table, not just on the submission of the trials and on the back end of patient enrollment with veterans, but also in prioritizing for us. So we've worked with a number of different organizations in that aspect.
No, thanks. I was just curious when you mentioned that that you were working on that population. It was nice to hear sort of what you're doing and the potential there to explore that. And the reason why I say that is really on how to decide on which projects to really go forward. And I think that's also what we're talking about in this first question. Is that you know, at least in CDRC, we're focused on real world data and then sort of moving that forward. But you know, where is some of this information coming from to be able to generate that real world data? And a lot of it over the past probably 5 or 6 years has been really this explosion in AI and really trying to go through a large list of you know, both the literature as well as other avenues to explore the data that's available already from a preclinical standpoint, and trying to predict the potential pathway. Well, knowing the potential pathway, but trying to predict what drugs could be used to address those. And I know Matt and Kaiser are here on the panelists, and they do a lot of this, and wondered if you know, how is it that at least for the pediatric portion that we can partner better with those groups that are doing that type of research.
Yeah, I think in response to the you know, the question of, you know, how can the real world data inform drug repurposing, and you know, how can that real world data push you know, a proposed treatment into the standard of care? I think that I would look to the example of what's going on in COVID now. Not that it's specific to these vulnerable populations, but I think it can show a nice cyclical relationship of real world data giving enough confidence to investigators to initiate a controlled trial with a relatively small population and limited effect size. And then using, once that becomes the option for care, using real world data to find other relationships between a drug exposure and outcome. So I think, you know, steroids for COVID is a great example of this. You know, it's only because of real world data from China early on that showed that patients were, you know, doing okay in response to therapy with steroids, that the recovery trial was able to perform an RCT to evaluate it. The same thing goes for, you know, tocilizumab. You know, several real world studies show that there may be some benefit in some populations. And then the REMAP-CAP trial narrowed in, did an RCT exactly on the patients of interest and showed a benefit. And then with remdesivir, it really is a repurposed drug initially developed for other viral illnesses. The ACT-1 trial showed some improvement in, association of remdesivir use and time to clinical improvement, but no mortality benefit. But then later, studies using the Premier database with 45,000 patients or so showed a mortality benefit. And so, I think the hope would be that we can use this sort of virtuous cycle for vulnerable populations as well, using machine learning techniques to identify subpopulations in real-world data sets that show promise of benefit of a therapy, and that can inform randomized trials, which can then inform bigger real-world data analyses.
And Matt, I completely agree. And COVID is such a good example for all of us because COVID is one of the first times in history that everyone came together to answer a problem together. And that in itself is one of the limitations and common barriers that we all face when you're defining these special populations, because depending on your institution, your region, there's variable definitions of disease. No one was arguing that COVID is COVID, because everyone said, yes, this is COVID. But once you have variable definitions of disease, and then you're trying to see the effect of therapeutics and repurposing drugs, that's where some of this limitations come in for all of us, where it's, okay, we don't know if this will work here because this is just regional, this is just institutional knowledge, this is just institutional experiences. And then this is where the CDRC and just the work with AI and machine learning, we can all come together. Is first, we need to harmonize the data, put all the inputs together. And this was done with the National COVID Registry, the N3C, where data was harmonized. So you could actually define a clonal phenotype, a co-blended type. And then you could look at the effects of the drugs. So that in itself is something where the CDRC community, we could work towards harmonizing their data inputs, bringing data together, defining a disease together, and then testing the drugs that we've already used in these special populations, because we have enough small studies, case reports, local institutional registries that have answered questions before. But everyone says that it's just a small n, so we don't take it as something that will work locally or for us, but together I truly believe we can answer more questions.
Thanks, Kaiser. Sorry, Matt, I'm sort of taking over a little bit, but this sort of goes in line with what Millie is doing at FDA. Millie, could you talk a little bit about CureID and that particular aspect of collating that information and making sure that it's harmonized so that a larger analysis can be done, and maybe some lessons learned in CureID.
Yeah, sure, Marco. In fact, I was gonna come in because I had a question for Kaiser, but that can wait. But just taking the conversation forward, Marco, you know that when we've entered into CureID and when we started to expand into special populations, we've been challenged by which disease to prioritize. Should we go for A? Should we go for B? Then there is the whole thing about safety versus efficacy. And this is anyways a niche population that we're talking about. And I was wondering, Kaiser, if you could talk about the neonatal severity illness index that you guys are preparing. Is that something that can inform the prioritization of the diseases in this population? Because that's something that we have definitely been challenged with. As far as CureID is concerned, it's what Marco was talking about. We have a mobile application, and it's also a website. And we go out and ask clinicians to submit their case reports to us, their individual case reports that they've had experiences with, so we can collate that data. But that's also a challenge. We know clinicians are very busy. And so on the other side, what we started to do was we got together and we started extracting literature from, extracting case reports from published literature. So we, of all the various infectious diseases that we have on our app, a lot of them we've done a thorough literature review of the last 5 years or so. In some diseases, it's gone even way past 5 years, and we've taken out those case reports, we've collated them in one place. We've done a similar exercise with clinical trials of those diseases. And the objective was that we can get and collate that data together. But what we are looking for here is not just collation of published literature, but where can we get more data from? Can we, can we go to small registries and ask them to partner with us? Can we go to institutions that specialize in special diseases and say, hey, would you be willing to partner with us and use our platform to collect this information so we can finally collate it and analyze it and look at some of the patterns? But that's what we are doing on our end using CureID. And my talk, which was about developing survey tools, was exactly that. Because when we started having these conversations, as I said, with experts who are experts from congenital CMV, we realized that before we even go up further to decide about and even start having conversations about a clinical trial, what we were hearing from everybody was that there is so much variability in the field. A particular institution, depending on how large or small is, they have a different, even though there are guidelines, even though there are recommendations by international groups on how to address symptomatic children and asymptomatic children, people are doing different things. And so one of the things we realized after having these conversations was maybe we need to go back to clinicians and ask them how do they address this disease? How do they measure their outcomes? What are some of the adverse effects due to which, because it happens to be a long regimen, that parents end up terminating the treatment. And so one of the big lessons in having these conversations was that there is so much out there, there's so much variability in the management of this disease, and we need to go back to the clinicians and get this information. Coming back to the challenge, when we started expanding into special populations, we were like, should we go for congenital CMV? Should we look at toxoplasmosis? Should we look at disease number 3? And one of the things our team has been challenged with and struggled with is how do we prioritize a particular disease? And, if you or if somebody else on the panel would like to address that, I think we'd all benefit from that.
Yeah, I'll jump in. And I know that we have a lot of expertise on this call with our colleagues from both Duke and North Carolina, where I know they've done some amazing work with regional warehouses and the North Carolina database, the Duke data warehouse. So I defer to them too, and I encourage them to answer. But you sort of hit the nail on the head in the sense that there is variability. And when you get a lot of people together, their agreement in itself at times is lacking for, one, defining the disease itself, then 2, managing said disease, and then 3, in terms of what is actually important that impacts that disease. And depending on where you are regionally or how you manage the patient or what's actually available, that in itself changes. Now, the approach that I've been taking and we've been taking institutionally is let's get all the data, because in order to do discovery-oriented research, hypothesis-generating research, we don't know exactly what will tweak things. Yes, what will cause the disease trajectory to change? What will cause your response to treatment to change? And we need to measure all of it. And that sounds grand because trying to measure all of it and collect all of it is a task. And that's where we leverage machine learning and AI to get all of the data all the time for all the patients. And then you actually have the opportunity to look at a specific drug, a specific intervention, specific therapy that you provide. Now, the idea of getting all the data all the time seems grandiose. That is possible. It is something that we can accomplish. We've shown this through the N3C, the COVID registry. We can harmonize all the terminology, and we can lower the activation energy in how to do it, because this whole process can be automated where you don't need an army of study personnel who needs to go and extract and spend hours of time with the EHR to pull the variables of interest. Is it perfect? No. But at the beginning, if you could get 70, 80% of your content coverage and you're making a pretty big dent. I'll stop there and I open it up to our folks at North Carolina and Duke who've been doing this as well.
Yeah.
Oh, sorry.
I don't want. I was going to jump in if that's okay. So again, I'm Matt Long, and really good to see you, Kaiser, on Zoom here. So I've been following your work, and it's been fantastic. So, you know, I just a perspective is that we a few years ago did something similar for caffeine, which was, and we used real world data looking at 100 infants from 4 different sites, and we sort of smushed all that data together, and it was a heavy lift to try to put those data into a manner in which it was palatable for us able to analyze. So any way that that people can get together to try to harmonize the data, as was mentioned earlier, really is very important, and something that came up, just a very minor point that took probably 6 phone calls and 20 or 30 emails was we kept getting the same time, it was a lab we were looking for, so it was like a sodium that was reported as the exact same time from the central lab. That was 2 different values, and they were close, but they weren't exactly the same. So it would be like 143 and 145, same time, same lab, same infant, same everything. And so we took a while to figure out like what was going on. And what happened is that the lab did an auto check just to make sure that the second value was the same. And if it was within a certain percentage, then they just reported out to the clinician the same, the one value. And that was just one tiny, teeny little thing that, we just had to work through, is that, when we got the data from the central lab, they gave us 2 values. The clinicians had no idea because they just saw what was reported in Epic. And so that was just one, minor little detail about things that can be really important. And so, I just think that, thinking about these things beforehand, and, it's sort of like we're sort of on the other end, is that the clinicians do the best job they can, including myself, in putting these data into, it's an electronic medical record. It's not designed for research per se. So anything we can do to try to sort of make it more palatable and easier to work with, would be great.
Yeah, and we had a comment in the Q&A box that I thought was appropriate to this part of the discussion, which said, in the discussion of real world data and its use as evidence, it is important to distinguish the data collection mechanism from the trial analysis design, as real world doesn't necessarily equate to observational. So registries have been used as data collection mechanism for prospective RCTs, which I do agree with. That's part of Cure ID, that we're not necessarily, we're collecting real world data and registry data, but it's not to directly have create an observational trial. It is to inform future RCTs for use.
But would you say that FDA now is open to utilizing real world evidence to be able to support labels changes that include obviously a sponsor that comes in with an RCT, or maybe with at least one RCT, but oftentimes more than one, in order to support that supplement?
I suppose I can't say that myself because I'm a fellow. I probably shouldn't answer that question.
Yeah, no,
But
I don't want to
terms of
necessarily put you on the spot either, Kate. But but it's really like I'm thinking about this more of the 21st Century Cures Act, and actually the second version, Cures 2.0, is currently in draft form and is circulating. So there's, I think, an interest from the legislative sort of branch to be able to help move this needle along, and it's not that they're going to say this is how you're going to do it, but it's obviously an interest to be able to help inform regulatory decisions, and that's a critical question, is that how can we use, and what are the sort of uh, the barriers or the issues or the challenges. Things like quality of data is gonna come into that. And as we all know, real world data is a little bit messy, but there are opportunities here that we can explore to be able to make it a little bit less messy. that's a potential.
Right.
Knowing the fact that there's always going to be challenges on this, but again, we're not hanging our hat on that, but we're really looking at it as a way to be able to support. Look, if this is what we found in a clinical trial, does this equate in a real world setting? And if that can support it, then that's exactly what it is. It's a support tool. And just wondering from
if I can jump in, because I know I'm not speaking for the FDA. I'm not at the FDA. Cures Within Reach clearly is not. So maybe it's helpful if a non-FDA person provides some perspective. We just a month ago had an event on real world evidence and real world data and how patients can engage in this process. And the moderator was from the FDA's real world evidence team. So they are absolutely aware, working on it, providing, figuring out, working with the ecosystem, working with researchers, and how you take results from randomized controlled trials and how you interact and add on other real world evidence case reports. They're very on top of this, and I want to give them credit where they may, it may not be a clear path, but they're very much working on it. And there's a whole mechanism at the FDA currently today in this aspect.
Right, and Benjamin just actually added in the Q&A that yes, this is exactly where I was going with this. Is that the guidance for using real world data just came out recently, and that's caused a lot of discussion, and that's exactly what it needs to do. And this is really just my way of saying that this is a great opportunity to start having these conversations, not only amongst ourselves, but to bring in regulators to be able to hear a little bit more about what their concerns are, what their challenges are, what the issues, and really start having that dialogue.
Marco, this is Prabha from FDA. I'll just jump in there too to say that I think one of the really critical aspects is to know what the goal is, because we can use real world data for so many things. It might be evidence generation to support clinical trials, like Millie had said. It might be augmentation of a clinical safety database that's limited, in which case some of the noise is not as important. Or it might be trying to do an effectiveness trial to build on maybe a small data set that supported approval, but wanting to know whether those results are more generalizable across the population, including populations that were unrepresented or underrepresented in the clinical trials. So depending on which of those buckets you're in, I think that that goal needs to be very clear from the beginning. And this is a place where FDA is certainly open, but still learning as well. And with learning comes caution. And so I think having those conversations early with those clear objectives in mind is really critical. And the guidance document that was referenced has a lot of those considerations nicely laid out for folks who are not as familiar with the FDA process. Guidances or how we communicate to the world, to try to provide information that is broad, that's not always very tailored to a specific product or maybe not even a specific disease, although we have those as well, but to provide a framework with which you can start in bringing those more focal conversations to the respective review divisions.
And Marco, this is Perdita. I was just going to actually chime in to follow up on what a few of the panelists just discussed. And in the BPCA Pediatric Trials Network program, we actually have submitted real world data, as Matt mentioned before, for our caffeine study and our ampicillin study. And for those who are in the audience, if you refer back to Dr. Greenberg's presentation, I think she talked about some of that during her presentation. So we actually have developed that paradigm to use real world data either to augment safety data or to support dosing data, and have had 2 successful, at least, label changes utilizing real world data. But what I wanted to sort of bring up really quickly was, and I think it goes back to what was said in the chat, was we have to really distinguish data from evidence. because the FDA, no matter what, is looking for evidence. I mean, as much data as you have, that's awesome. But if you really can't translate that into evidence and have clear data quality, that data can be used for evidence for label change, et cetera, then the amount of data you have, it's not irrelevant, it's important, but that quality must be there. And so there must be some sort of upfront hypothesis-driven data collection that's important for, if we're going to the FDA to use this information for label change in particular, we have to have that quality up front. And whether that data is extrapolated from the EHR, you know, and put in some platform that improves the quality, maybe, I don't know. That's one way, but there has to be some way to improve that for what we want if we're really going for label change.
So I think this is an interesting conversation that has been sort of talked about both before and at all other sort of sessions, as well as these objectives. So if we want to, you know, we really do need to see the end goal in mind, pick that end goal, and then start working backwards to say, you know, what is the information, where is it, how do we do that, and then we sort of make this roadmap of getting to where we are now to where we want to be. And that's the questions I think that in this group we've started talking about. One is the congenital CMV, another one is bronchopulmonary dysplasia, and the question that I have is what is that end goal? So yes, we do want to see the potential of using drugs that are currently approved and available to be able to treat patients that will actually be efficacious and safe. But we need to go all the way to a you know, is this just a requirement to say we just need guidance documents, so we just need evidence for guidance documents, or do we need actually a label change? And that's what I was going to sort of ask from the clinicians in the group is that, you know, from your perspective, what do you see as being something that drives it? I know the BPCA does do that, but I'm completely, you know, I completely agree. I think that a label change is important. But from a practicing clinician, is that going to be something that drives it, or does that label change then also drive guidance development and uptake and things like that? Because I've heard it from both sides, and I'm trying to understand it in different contexts, whether label changes are actually what will drive clinical practice.
Marco, this is David. If I could jump in with my opinion on that, I think as pediatricians, we're all very comfortable with using medications off label. I mean, that's just our reality. It's not necessarily a desirable reality, but that is kind of where we are and where we've been for forever, I guess. It's getting better with the Best Pharmaceutical in Children's Act and so forth. We're making movement there, but it's still part of what our comfort level is, maybe to a greater extent than other physicians taking care of other populations. That said, I think that guidance documents and guidelines are very important. I think it's even better when those recommendations are reinforced with a label indication. If I had to choose one or the other, probably the guidelines have more, in my judgment, more real world impact. People look up for IDSA, they look for, you know, whatever the group may be that's putting out a guideline, Pediatric Infectious Disease Society. They look for the evidence and the way that the process was put together, but mostly they want to know what to do for that patient right now. But when they can do that and have the label indication, so they know the dose was, you know, has sort of received that official blessing, if you will, from Food and Drug Administration, that makes it an even easier decision on the part of clinicians.
David, any thoughts about going to those guidelines to be able to sort of say, you know, I'm also trying to think about, you know, going beyond congenital CMV, like to be able to take a look at these guidelines to say, you know, where is the gaps? Where are the gaps in here? You know, there are oftentimes, and it's all over in pediatrics, where it's just you know, basically physicians and groups that sort of say this is how we normally do it, but the level of evidence in that is actually very, very small, and that would you know, obviously be great if we could add additional data, whether it's real world data or something, to be able to sort of say, you know, let's elevate this level of evidence a little bit more and see if there's something here or not. Is that, I guess, a way that we should be thinking about, you know, projects and how to prioritize things?
Well, I'm not sure if this is going to be responsive or not. I think that it is easier to develop the guidelines and guidance documents in the first place if there's an indication. So in other words, in the case of congenital CMV, the European and the international guidelines and American guidelines as well through the American Academy of Pediatrics, they're out there about who to treat. Those were developed without the benefit, you know, in the United States of an indication with Valgan or ganciclovir for congenital CMV. If that indication was there, it'd make it even easier to develop the guidance and or modify the guidance and guidelines as needed. And as we know, and is appropriate, the guidelines and guidance documents are reviewed at set intervals depending on what the organization is, as to what those intervals should be. So I think they do interact perhaps to a greater extent than maybe my initial response would suggest, depending which is coming first or whether they're both happening at the same time, or if licensure indication occurs in advance of a revision to a guidance document. But as I say, I'm not quite sure that's getting at what your point is through the course of your question.
Yeah. So what I'm basically saying is that, you know, do we move in one direction or the other? Do we sort of go through the guidelines first, or do we go through the regulators first? And that's what I'm trying to see, is that what's the biggest bang for the buck for individuals that are currently treating.
So I have a comment about that, actually. Sorry, I didn't mean to speak over.
Go ahead, Matt. I'll go next.
Okay, so we actually did something similar to this several years ago, where we evaluated. We did a sort of, it was somewhat informal, but it probably holds true today, which is if you ask pediatric residents at UNC and Duke, you know, have they ever read an FDA label? The answer was 100% no. And if you ask most people, they probably haven't. And so, but, and here's the but part, the good part is that the label, most of the residents and even in Epic now and the pharmacists have pediatric dosing guidelines or handbooks. And so those handbooks and those guidelines, and there are a variety of them that are out there, use the FDA label as their primary source of information. And so, you know, I think Rachel was going to say something about that, but my understanding is that once a label change occurs, then those handbooks are pretty good about picking up that change. And so, you know, for example, caffeine changed. Perdita mentioned that. There's some antibiotic changes that the Pediatric Trials Network has done, and so it trickles down into clinical practice relatively quickly, and the guidelines pick up that as well. And so, what most guideline groups do, whether it's the American Academy of Pediatrics or the Committee on Fetus and Newborn, is they try to do a systematic review where they evaluate whatever the condition is and what the conditions are, and then they have a level of evidence about what to do with that. And those things include, you know, the FDA label if it's about drugs. And so I think personally, my opinion is and experience is that it is very important because it has a wide range of dissemination with the FDA label, number one. And then the other thing to think about is that the FDA, US FDA, serves as the regulatory guidelines for a variety of different countries. So although we're focused on the United States, I believe mostly on this call, it has even wider range and broader range.
Yep, I agree with what's been said. Definitely, I think clinicians are, you know, we'll go to guidelines and we'll go to handbooks for dosing, but ultimately, the FDA is there for a reason, and that's to be able to serve as the authority on what's safe and effective. You know, because of the way drug development happened, you know, unfortunately, children were left out of that process for a long time. And so we're stuck in the situation where children, we're playing catch up with children and other special populations, especially infants. You know, it's shocking to most people, to most clinicians, actually aren't aware of how many things that they're prescribing are actually off-label. If you want to treat pain in infants, there's almost nothing that you can use for that's according to the FDA label for some of the most commonly used drugs in the NICU where I practice. But I don't think that should stop us. I think we should aspire to have the FDA continue to be that ultimate authority on what is safe and effective, and to gather the evidence and submit that evidence so that you know, it can be widely disseminated. We've shown in one of our studies where we did some more phase 1, phase 2 studies to look at pharmacokinetics of a couple of what we've done for many drugs, but for a couple of antibiotics, looking and then looked at, actually used real world data to look at the uptake of those recommendations based on the phase 1, phase 2 studies. And when the study was released, we saw an improvement in the appropriate dosing of those drugs, but it wasn't until one of them had a FDA label change, we actually saw that uptake kind of come up further. And so I think, you know, that's at least one piece of evidence to support the importance of label changes. And I think aspiring to the ultimate goal of having just as much data out there to treat pediatric diseases as there are for other populations.
And if
this
I
is
could,
Prabha. I just want to jump in again too, and I'll be short, David. I'll hand it over to you. But
I was just going to say I agree with that.
I agree with that too, and I think sometimes it's helpful to learn from a positive example where things go well, which I think in the field of pediatric ID is HIV. So we have drugs that were developed in the time primarily after we had pre-N BPCA legislation, where you can see how labeling and guidelines can be very complementary and very timely and very reactive to being espousing what the ongoing improvements in standard of care are. So I serve on the guidelines panel for HIV. And so I see both sides, and there are times that the guidelines say something that is broader than what the label says, because labels treat drugs one-on-one. Like, did this drug meet the standard of safety and effectiveness? Yes. Does it say anything about which one you should use? No. And that's why the guidelines, if they have that complete information of the drug's activity, safety, pharmacokinetics, and whatnot, then the guidelines can use that information in a broader context to prioritize what you should use, to optimize regimens. So a long way of saying, I don't think these are mutually exclusive sources. They need to be complementary sources. And if you can generate those data in a timely fashion, then this process works most optimally. Sorry, back to you, David.
Well, and the only additional thing I'll add is kind of your word timely fashion or words are building on what I was going to say next. HIV has a lot of money in their system, and there's a lot of resources that can be applied. Something like congenital CMV does not, and so that I do think that we need to be mindful of the limitations and yet the major unmet medical need out there. And the limitations can be both resource limitations, but also time limitations. These are rare diseases. We're talking about special populations today, and I know y'all have talked about rare diseases before, but congenital CMV lives in both of those spaces. And so it'll take a decade to do, or maybe close to a decade to do an important trial. And so what we're left with is what we're left with. I mean, the studies, hopefully in congenital CMV, tell an important story, but they'll leave questions unanswered, and they'll have gaps in the data sets or in the interpretation thereof that are a reality. And yet, if you look at the totality of the information, my feeling is, if you look at the totality of the information, the story suggests, benefit of treatment in a population, in a group that is symptomatic and can be started within the first month of life. So I do think we need to be a bit forgiving in the way we look at some of the individual data pieces to put together what is the full story, the full composure of the photograph or the picture that they're coming together to tell.
David, I completely agree with you. One of the things that I wanted to, first of all, with this conversation is to make sure that we're all aligned on what our goal is. And I think that we've come together on that for sure. I just wanted to make sure that we had that conversation, that we're all aligned on that. And I think then obviously we are. But the next question is RTC design. So, I think because the children and neonates especially have not been really put on the forefront of that, and it's been an afterthought. And so obviously neonatologists are basically saying, well, we're going to have to treat, and this is what we're going to do. So there is sort of this and I think Rachel has brought this up for CMV, for congenital CMV, is this loss of equipoise. How can you then refuse or have a control group where you're not treating a patient if you know that that's a standard of care, for example? So you can't remove standard of care in that case. And so this becomes a sort of how do we design a clinical trial? Do we answer the question and do it ethically and morally? So that's one thing. And then I guess this could also start dwelling into some of the chat discussions that we've been having on data quality, and just wondering what other people think about data quality and how we can best address some of that moving forward. I know, Perdita, that you and Kanwaljit were talking a little bit about that. I think Matt Robinson, you obviously are developing this tool to be able to extract data from electronic health records. So, what are some of the lessons learned or potential ways in which we can work together to ensure that we have a better quality of data coming out of real world data sources?
Maybe I can just jump in. And I know we're talking quite a bit about neonatal and certainly pediatric populations, but maybe on the upstream side of that, which is pregnancy and during this gestation, looking at effects, our team has tried to answer a lot of these data quality questions within the EHR. So I think Kaiser brought up N3C. We work very closely with N3C through the pregnancy domain team, very much focused on how can we build ways to better vet real world data sources to then be able to generate hypotheses that have higher translational potential for clinical trials. And that can inherently inform the design of the clinical trial in terms of looking at the appropriate types of endpoints, looking at the appropriate types of drug disease corollaries that need to be investigated. And of course, when you're mentioning cases of lack of equipoise, looking at the extent to which the EHR can be used for things like target trial emulation, which is where we may be able to essentially simulate the elements of randomization and controlling within a real world data set. And so I think, maybe this ties back into the first thread of conversation about, where we can innovate in real world evidence for this group. Certainly kind of within the pregnancy space, there's a big problem in terms of even being able to answer fundamental questions about how we describe pregnancy. I think Perdita brought up a great point about common data elements. And this is something that through N3C, we've done a lot of work, is to actually define in the EHR, how do we know when somebody's pregnant? And how do we know how do we know gestational timing? Because these things are not inherent to the common data models that underlie most EHRs. And in order to answer treatment questions, in order to do target trial emulation, we first, of course, need to be able to phenotype the population. So I think, there are two angles to coming to these types of questions. There's the very important regulatory kind of policy type questions that can inform what we may be able to do once we have evidence to be able to push through. But then on the fundamental discovery end, we just are so far from being able to do end-to-end studies within real world evidence sources, because we can't even tell when somebody's pregnant. And then when they're pregnant, we can't tell what the gestational age of the fetus is. And then, we also have ascertainment issues in terms of chronic diseases, for example, during pregnancy, being able to reliably have those coded within the EHR. So I think there's a lot of work that needs to be done in terms of just answering fundamental questions of data reporting. And I would argue that this is not completely a computational exercise, that we can't simply use machine learning or other forms of pattern detection to be able to do this, because while machine learning can help us to identify structured data elements within the electronic health record, it can help us to identify from an analytical standpoint the patterns within the structured elements. To actually be able to have the data source reflect real world clinical knowledge, there has to be some type of Delphi process that gets baked into how we actually go towards defining these data elements and then applying them into inferential studies. So there is some human machine interface that we have to find kind of the right balance between the two different modes of learning. But I think it's very dangerous to think that we'll be able to achieve end-to-end automation that can then feed into enhancing the likelihood of clinical trials. In reality, enhancing the likelihood of clinical translation from these data sources will require even more concerted effort between humans and machines to get to those data standards, those data quality elements that everyone can agree is acceptable.
Add something to what Anup is saying for the group to consider, or at least ponder in this, and that is connecting the pediatric and maternal health community with what I had mentioned earlier on the diversity, equity, inclusion, and representation of these trials and of the realities. For example, and the data I'm going to share is just Illinois, but I imagine the United States is very, very similar. Black women, actually Black babies in Illinois of very low birth weight. Black babies are 3 times more likely to be of very low birth weight. And Black babies of low birth weight are 2 times more likely to be Black babies than the rest of the population. And there's a real opportunity to add to the clinical trial and real world evidence, and the conversation we're having is inclusion in all of the populations for these trials and all of the opportunity to include everyone, not just in trial design, but enrollment opportunities, in access to maternal health. Like you said, Anup, we can figure out who these people are that are pregnant and do something earlier to try and impact on the health inequities going on. And I just wanted to add that to the mix.
Hey, this is Jenny Taylor. I just wanted to speak up at that point because I do think that's so important. And as part of our neonatal global rank score development, the first one of the first steps has been to do a modified Delphi consensus process, and half of the participants have been parents and NICU graduates, and being able to hear their voices as we think through which endpoints are most important to include and how to rank them by severity is just so helpful moving forward. And we've been lucky enough to kind of utilize our research assistants locally, as well as parent groups to think about diversity and to design our surveys in such a way that they can be accessed by people who don't have computers and just use their phones and use technology that way. And so thanks for bringing that up. I just think that's so important.
Yeah,
Go ahead.
Well, I was just going to add one other challenge. We've talked about the challenge of making sure that automated extraction of data from EHRs represents what's actually happening. But the next, let's say you get that just right. If we actually want to use real world data to be evidence, we have to be very careful of many additional biases, such as confounding by indication. And this is a huge problem in using this data for drug exposures. This happened early on with COVID. If you take a bunch of healthy people who have COVID and give them any drug, it can look like this drug is great for preventing severe illness and death. So assembling the right populations to compare who have received drug exposure and who have not can be really challenging. And the methods that one uses to do this are, subject to incredible bias. It's very easy to search through several different methods and choose the one that leads to the outcome of interest for your study. So it's just as we approach this idea of using large data sets and using this real world data and turning it into real world evidence, you have to be very careful of these biases. And that can include in finding special populations that seem to benefit from a therapy. Just have to be very careful that these assumptions that we're making hold up to additional scrutiny.
And I was just going to echo that where Matt's saying it, Anup is saying that the ability to extract data just doesn't necessarily mean that AI, machine learning, is just going to take everything, do everything for us. If anything, the man and machine combination needs to be better. The person and machine combination needs to be better, where you need to be very thoughtful, because you can, when you have this much data, show associations, show relationships that are just there. But we have to be thoughtful in how we go about doing this work, because it's very easy to get lost within just the numbers and what you're putting in. And then Anup's point, in terms of something as simple as gestational age for pregnant women is not available. And then we're talking about drug discovery and drug repurposing. Something as simple as drug dosage is not available in some of these common data models. And when you extract this data in itself. So when you want to rely on machine learning AI, the output that you're most concerned with isn't available. So in terms of developing that and creating that bond and creating that synergy of getting the right data in itself is something that takes time, because coding is only as good as how you code it and what you code for.
I'm wondering, Kamaljeet, whether you could chime in on processes that that you've been using to be able to address sort of that question on getting that right data, ensuring the quality is correct even before you do the analysis.
Thanks, Marco. This is sorry, let me just turn on my video because I had turned it off. Apologies. So yeah, this is a complex issue. As I was discussing with Perdita just a few minutes ago, how exactly do you decide whether a piece of real world data is actually of sufficient quality or not, right? And as she answered, having a common list of common data elements is a good start, and that is exactly what we do. We whenever we're for a lot of our projects at International Neonatal Consortium, as I said, that we are working with collection of large amounts of real world data, whether it is electronic health record data or registry data or other types of data, to create a database that we're hoping to use to answer some of the, I guess, unmet need questions in the neonatal space. And what we really do is we start with having it's for us, it's basically kind of a two-way street. So we provide each individual institute that we work with a list of all of the potential data elements that we're interested in, and then we ask them to send us basically their data dictionary of sorts, in which they will list us in their electronic health record system or in their registry what are the specific data elements that they have available. And we basically do a kind of a cross matching between what we want and what they have available, and we repeat this process for all of the subsequent sites that we work with. And our, I guess, even I understand that even this process is not going to be 100% effective in ensuring that the data that we get from each individual site is going to be of a sufficient quality. And this is just the reality of it. But our hope is, by doing this process and working with each site, having quite a bit of back and forth, and basically trying to get a version version of data that we think is clean enough for our purposes should work. We actually haven't really gotten to a stage yet where we are in the stage to evaluate the data quality, because we're still in the data collection phase as of now, because if you do not have any data, you certainly wouldn't be able to say what quality of the data is. But this is the process that we are using in our projects here at INC.
Thanks, Kambaljit. I just, I'm just looking at the time. I can't believe it's an hour and a half already. There's so many other questions that I have, but I wanted also to turn it over to Jason. I haven't heard from Jason today. And as we were starting to talk about, these different aspects, we brought in congenital CMV, but the bronchopulmonary dysplasia is also a project that Kamaljit is helping pull together, again, using real world data, electronic health records. And we're wanting to be able to collaborate more effectively and being able to share that data more widely. But on your aspect of using Montelukast for this, I think in your presentation you talked really about dosing and safety. And I think that those two things are going to be critical, but I would like to also put a plug in for efficacy as well, because we really do need to make sure that what we're doing is going to be effective for that treatment and to be able to help the babies that potentially can do that. Any thoughts about efficacy and that inclusion in sort of in future prospects for that aspect of it?
Yeah, thanks, Marco. I know I've been quiet. There's so many issues around EHR and data quality. In terms of Montelukast, I mean, it's interesting because that's a topic that involves both of the conditions I'm kind of interested in, asthma and BPD. And those two treatment domains are very, very different in terms of what you can view through the EHR. So BPD, babies who are developing BPD are in, they're a captive audience, so to speak. We have a lot of data, and analyzing the EHR becomes sort of a higher level of complexity. It brings on a lot of additional levels of complexity. In the world of asthma, the EHR is almost blind. When I think now, I back up and say, if you're talking about asthma in the ICU, asthma of a hospitalized patients, I think it gets a little bit easier. But where sort of the holy grail of asthma is sort of preventing chronic symptoms, preventing exacerbations, and the EHR is not very good at knowing drug exposure because of the outpatient setting and a whole host of additional challenges. Are the medications being refilled? Are they being taken? Are there adherence issues? So our ability to assess drug exposure towards asthma outcomes is very poor. The other additional component, just outcomes of asthma. So pulmonary function is rarely captured in the EHR. Event capture, daily symptoms. Again, I'm sort of going on, but I think that if asthma could be, asthma is by itself important because it's the most chronic, most common chronic disease in kids, but I think it also represents a lot of other or it's an example of other chronic diseases in pediatrics where we probably need to focus specifically in this area. One of the thoughts that was going through my mind is that the tension is that in some ways it could be seen as a privacy concern, but on the other hand, I think it also could be really a patient advocacy concern, that patients with certain populations, these new technologies were really not able to help them in coming up with new and better treatments, say for asthma or pediatric hypertension or other things. Now, I know you asked about Montelukast. So I think the efficacy in what we're proposing is very important. It is a secondary outcome. And I think that that's because what we want to know first is it safe in the BPD and the infant population? And we also want to know what the best dose is. So, one thing I've learned from Matt Lawn is that there are a lot of phase 3 trials that are looking at, is a drug efficacious? And we expend millions of dollars to determine is a particular drug at a particular dose efficacious. And too often these big trials, these expensive trials fail because we really don't get the right dose. So that's why I guess we make a plug for having more phase 1 and phase 2 trials where we're really looking at the best dose based on what's safe and what appears to be effective. So that's why I proposed in my talk taking that approach. I don't know if that answers your question or not, but
it does, absolutely. That's exactly what I wanted to hear from you, because it's exactly right. The last thing we'd want to do is spend time, money, energy on doing a large clinical trial on efficacy and then figuring out, oh, we got the dose wrong. So it makes complete sense, and it's something that potentially we could work together in parallel.
And that sentiment is completely logical and makes a lot of sense, and yet it's those big flashy phase 3 trials that keep getting funded. And because I think the phase 1 and phase 2 studies are totally necessary, but not exciting and not innovative enough to pass through some study sections. So I think one challenge we have is who's going to do those and who's going to fund them, so that we can eventually get to the phase 3 that we really need.
And I guess, we've been talking a lot about electronic health records and the challenges and all that, and whether even we have the right data there, absolutely. And it's, I don't want to get completely hung up on EHRs and electronic medical records, because there are other sources of real world data that we can also start exploring. Any thoughts on that from this group? Things that have happened in the past or other projects that you've been involved in where you saw that that being beneficial, having this additional sort of data set other than EHRs?
I mean, I think that that's a great point. There are a lot of other sources of real world evidence for hypothesis generation. Patient registries, for example, are a good source. In the special populations community, though, particularly around pregnancy, a lot of these registries are not open source. So it's very difficult to gain access to many of these registries. And the ones that are open source tend to be quite small. And the other difficulty is that a lot of resources that are maintained by patient advocacy groups, while certainly great that they engage patients in being able to directly source this information, there may need to be increased incentivization for those groups to publish resources in structured formats that are amenable for computing. Because to pull in all of these data types that you need to power efficacy predictive analyses, for example, it's important to have a good database. And if you only have 500 or 600 data points when you're working with questions that require n in the thousands, then you're inherently setting yourself up for an underpowered study on that basis. Another good source, claims data from insurers or from public payers. These data types tend to be very large, just given the catchment for insurance companies or for state-run or nationally run insurance programs. And it tends to be actually that these data are quite well maintained. So companies usually have a very good way of maintaining their claims data that creates amenability for computing. Difficulty, of course, is incentivization for private stakeholders to participate in this type of research, creating the right value proposition. Maybe that would be reduced healthcare utilization costs for a more efficient treatment strategy. But being able to really convey a good line of reasoning there to bring in the right payer advocates is certainly challenging, and it leads sometimes to a difficulty in being able to work with these types of data sets as well. Another, I think, infrastructural problem in being able to source claims data is the necessity of a distributed data network. So the idea that there are certainly privacy and security challenges to exporting terabytes worth of data from insurance companies to academic sites. So methods that are developed to probe these types of data sets need to be reproducible enough to work within different computing environments. They need to be amenable to the people who actually hold the data. So for example, anything that requires a use of cloud technology would not be appropriate for many insurers because they aren't comfortable uploading sensitive patient information to a cloud and working over those types of data. So there are a lot of capacity building avenues that still we haven't explored in terms of how to get the most value out of these data. But it's important to really keep that stakeholder engagement piece in mind to help advocate for the needs in the investigative community about how we can best use these data types to inform clinical studies moving forward, and developing the right value propositions and communicating them is very important in that way.
This is Prabha again from FDA. I just want to build on what Anup said. You raised a lot of great points, and just bringing this conversation back to congenital CMV, one of our session topics. We are actually in the division doing some work with the Sentinel system, which is FDA's distributed database that mostly partners with private insurance companies, have a very large encatchment area. So we are trying to use claims data to look at congenital CMV. The advantage here being you have a gigantic pool of participants and then can look across a lot more places than you could if you were going site by site. The trade-off, of course, being you lose a lot of sensitivity in the type of results you can abstract. For example, I can look to see if somebody had a CMV assay ordered, but I can't see the result. But if you're just really trying to capture patients, then it's powerful. And so, and this is something that I can justify as part of FDA's due diligence in advancing drug development. So I think partnerships here are powerful. So if government can help do some of the work with tools that are available to government to do hypothesis generating work, then that can set up academic groups and industry to carry that work forward. But the critical thing being that we all have the same goal in mind.
And I'll add to that albeit a different population altogether, I mean, still congenital CMV, but a different group, in other words, to work with. The patient advocacy groups are well established with congenital CMV. We have the National CMV Foundation here in the United States, and then they interface with similar parent groups in other countries around the world. And these are very dedicated, very devoted families that are working with academia and others as well to try to advance their own cause, which is to try to raise awareness of congenital CMV and diagnostic opportunities and therapeutic opportunities for congenital CMV. And I'm not saying they necessarily would be absolute partners in these kinds of endeavors, but if their focus overlaps with where our needs are, I know that we will have a great partner in them to be able to develop some of these kinds of, if we're looking at a survey or we're looking for, opportunities to characterize better what the outcomes are in the real world for children not involved in therapeutic studies, for example, or natural history follow-up studies and so forth.
Thanks, David. In actual fact, we initially connected with Gail Harrison over at Baylor, who has the Congenital Cme Registry that she's been running for the past 40, 45 years. So, and they're very interested in partnering and seeing how they can help. So I think this is an opportunity to bring in people that are doing registry work. I completely agree. The patient population, the advocacy groups need to come in. The potential of getting government to come in, potentially with Sentinel and other resources to be able to assist with this. It's starting to become something that the groups can now start coalescing on to be able to address, again, that one big question that we want to get to. So I see that this is going really great. I haven't approached claims or public payers as of yet, but it's something that we may want to do in the future. I think that may be an opportunity where we can explore a little bit further. I know that we've got maybe about 15 minutes left. I do want to give some time for others to ask, but I think, Millie, the other question that I had is the work that we're doing on surveys, and rather than going through the clinical aspect of this, but really taking a look at more of the patients and the advocacy groups and the parents, especially for congenital CMV, to be able to discuss their experience in this process as well, and what they could bring to the table.
Yeah, absolutely. I think, Marco, that's very essential. We've always talked about the fact that while clinicians and getting information from clinicians is a small part of it, the population that is most invested in these children are the parents or the caregivers. And it's like going to them and asking them for the information, and they are the most invested. They are the ones who will be assuming, and others can chime in, of course. Our point always has been that they are the ones who are invested, and they are the ones who are going to return to the app and answer those questions that we're asking them. And I think that's something that we really must tap into. I know Cure ID has done some work, and it's in progress for long COVID, a survey for long COVID. And if we can use the same kind of technology or strategy, we've already have been in conversation with Gail Harrison, and she has committed to us that their group knows other groups, other patient advocacy groups that they work with, and they're willing to disseminate that survey for us. But I absolutely think that asking parents or caregivers to give us that information can be the most one of the best things that we can do to gather information. But if you'll also allow me, Marco, I had a question for David, and maybe Prabha can chime in too. David, your presentation was very interesting, and we've always known the difference between the symptomatic kids and the asymptomatic kids, but the way that graph moves in your presentation, it kind of blows your mind that 90% of these kids are asymptomatic, and as the age progresses, you see the progression of the hearing loss. And my question to both you and Prabha is, do we even have a plan for this subpopulation, for these asymptomatic kids? Do we have a plan? What are we going to do? What can we do? Are there groups out there working with this population? I'm very curious if both of you could speak to that. Thank you.
Happy to do so. Thank you for the chance. We did have a treatment study for asymptomatics. Very expensive study, because in order to find the asymptomatics, you've got to screen, and you got to screen 200 to find one. And so it took a lot of resources. We ended up closing that study down early because of a safety signal. There was a degree of neutropenia, which probably would be quite well justified in the symptomatic population. But when you're talking about asymptomatics, who most of the time are not going to have hearing loss, then at least with in our experience with valganciclovir, that was not a population that was where we felt that could justify continuing the study. So that study was closed, and thankfully NIH has allowed us to repurpose many of those dollars to start what I alluded to very briefly in my opening statement, is a biomarker investigation, with the goal being, if we could find a way to better identify the subpopulation of asymptomatics, and I guess symptomatics as well, but asymptomatics would be in response to this question, that are more at risk for hearing loss, then the risk-benefit ratio to study an antiviral drug in that population changes somewhat. So we've shifted away from a straight therapeutic study, which I would have loved to have seen all the way through, but it just wasn't meant to be with this particular drug anyway, and gone more toward, can we identify who's even at greater risk, and therefore future studies focus in on that subpopulation.
Millie, I don't have very much to add. I'm really happy to hear that David is doing all this terrific work. I mean, the resources, these are very resource intensive propositions, and in the setting of specific CMV therapeutics, it's something that we try to engage with companies in as they develop their pediatric study plans. And, you know, we can try to provide incentives through BPCA for them to conduct these kinds of work, these kinds of studies, but it's not something we can necessarily require. And so there's still a lot of work that needs to be done to determine how to make these studies doable, especially over the years of follow-up that are needed to get an interpretable data set at the end.
You, both of you.
And I see we have maybe about 9 minutes left, so we've got to wrap it up. And I was just wanting to sort of pitch this out to anyone in the group, whether you have some last-minute comments or thoughts from what you've been hearing over the past, what is it, an hour and 51 minutes.
Marco, this is Prabha again, and I feel like I've been talking too much, but I just wanted to say thank you for assembling this group, because I think what we can all do together is something terrific that none of us can do individually. So thank you for being the hub and bringing all these stakeholders to the conversation. It's really been a pleasure to learn from all of you, and I hope we can take this conversation forward.
Thanks, Prabha. And we'll be needing to expand this group. You know, obviously there's a large interest on congenital CMV, which we will want to move forward. There's you know, with the collaboration with Inc. and potentially with Jason looking at bronchopulmonary dysplasia as well, and seeing how we could help move that needle forward as well. I think we're open to other avenues, and one of the things that just recently came up, and this will be talked a little bit more in the next session, is going beyond FDA. Although FDA is great, we want to, you know, obviously work with FDA to be able to get some of these, but we're also starting to think more globally on some of these issues as well. And how do we interact and engage with other groups outside of the U.S. To better ensure that we're aligned, we're harmonized, we see what they're doing and how we could fit in. And so, for example, the EU and EMA just released a way in which groups can come in that are not sponsors to be able to do a label change at the EMA. And so this may be an opportunity, and I've talked a little bit about this with both Matt and Rachel and Jason about and potentially, you know, Perdita. You know, there are resources that are currently being used to get a label change at the FDA. If there are equal opportunities and parallel that are offered by other stringent regulatory authorities, you know, would it be useful to start taking a look at participating in some of those discussions as well? And it's just, you know, one way in which we can get a better understanding of what's happening on a more global level, and being able to not only just have this community within our national border, but, you know, outside of that as well, and participating in those conversations, because in some cases, you know, they're doing some pretty amazing and innovative ways in which to explore both the real world data as well as repurposing, and it makes sense to be able to collaborate as much as we can more effectively, not only within the US, but outside the US.
Marco, just want to add to, you know, what you said, shamelessly plugging ourselves here, that we really want to expand the special populations sub-projects. We want to build more sub-projects that can benefit both the pregnant populations, the neonatal populations, and some other, of course, special populations. So if you have an idea, and most of our work in Cure ID has been based on successful conversations and collaboration. So if you have an idea, please, let us know, come talk to us. Even if it doesn't go anywhere, we're always happy to get onto a meeting table and have these conversations and share ideas and brainstorm and see if we can take it somewhere. So thank you so much, Marco, for putting this together. This was great. And thank you to all the panelists and the audience listening in.
Right.
Yeah,
And
I'd like to echo that. And I was just gonna ask if Pam or Kitty or Chandler have the email address that if any of the attendees would like to work with us in any of our subcommittees on any of these projects. Like we're welcoming everybody, and we have you can look at the CDRC website, I know, but was there a specific email? I thought there was like a slide with some information.
Yeah, Kate, I was just going to say about that is that if some of these issues are something of interest and pertinent to other participants or people that are on this call, feel free to reach out either to me personally or to cdrc@cpath.org. And that was a slide that was flashed out right at the beginning. And we're happy to start having some of these discussions and bringing you into the loop and seeing how we can all work together, because that's really what it's going to take. It's I think that was said right initially at the beginning, is that it's going to be a community that comes together to be able to pull this. Not one individual or group can do this, but it's going to take this group coming together, and each one of us will have different strengths and weaknesses. So, getting that data in, the informaticists, the people that are working on EHRs, the people that are in registries, patient advocates. Then we need the clinicians to be able to really start thinking about ways in which we could utilize this data to better advance clinical trials. What are the questions that we need to address? Sometimes we have to go back to the drawing board, as David is talking about, looking at a biomarker, especially for those asymptomatics. So I think that there's a lot of efforts that we can do, but we need to come together as a community and drive it towards that main goal. So I think that's the next sort of aspect of this is ensuring that we are all aligned on that goal, defining that goal, and then start making a roadmap about where we are now, as I said before, and where we want to be. And then start plugging away at each one of these various stages in order to get to the goal that we want to eventually achieve.
Marco, if I could build on that, I think that where you and CPATH and these partnering organizations have such a critically important position to occupy is exactly this interface. I mean, when I think about what I would like to do in terms of additional studies, or what we've accomplished in terms of previous studies for the treatment of congenital CMV, I hope anyway, I have a relatively broad view, but I'm still pretty focused on an academic kind of assessment. Can I get funding for it? Can my partner academic institutions get the subcontracts or subawards in place? I mean, just the practical aspect of that. And it would benefit, undoubtedly, it would benefit all of our studies, in my judgment, if we also had at that early stage the opportunity to have some sort of interface and discussion and dialogue with FDA and with other stakeholders to say, all right, now let's assume this actually works. Can it then get what everybody else needs in order to make additional decisions on guidelines or decisions on indications or whatever it may be? The more opportunities we have for, it doesn't have to be binding dialogue, but just dialogue. I think at the very beginning or near the very beginning, I think the more chances we have to do that, the more opportunities we're going to have in these special populations and rare disease groups that take a long time to do studies in, to really have a chance to have each study that's conducted hopefully move that ball forward down the field.
Thanks, David. And one of my mentors has always said, it's not the boulders that you trip over during your path. It's all the little pebbles, and sometimes we just don't see where those pebbles are. And that's what I think the power of bringing in various groups with these diverse experiences can help us align and alleviate some of those, or at least illuminate some of those challenges that that we may not be thinking of, you know, at that early stage. So I completely agree with you. It's these early stage development discussions that that will be really beneficial and will pay dividends in the future. Okay, it's the top of the hour. It's, as I predicted, those 2 hours have gone by really quickly. More to come. This is just the initial volley, the initial discussions on getting this moving forward. Much more discussions to come in the future. I thank all of the panelists and participants that have joined us. And again, if you're interested in continuing some of these conversations in conference calls, please feel free to reach out. Thank you, everyone. I appreciate it.